Good morning, students. This class will discuss about what is sequence alignment, what are the methods of sequence alignment, different types of sequence alignment. What is sequence alignment? As you all know, when we discussed about different applications of bioinformatics, sequence analysis is one of the important applications of bioinformatics. What do we do in this sequence analysis? We compare the sequences in order to find out the regions of similarity and dissimilarity. So sequence analysis is nothing but comparing the sequences in order to find out the extent of similarity and dissimilarity. The method by which the sequence analysis is carried out is sequence alignment. So what do we do in this sequence alignment? We compare two or more sequences by aligning them in order to find out the relationship of these sequences in terms of similarity or dissimilarity. Look at this picture. Here we have shown the alignment of two sequences. I mean hypothetical two sequences, sequence A and sequence B. These two are aligned one below the other. When these sequences are aligned, we will have the columns. Columns are nothing but the vertical arrangements. We align the sequences in such a way to get the identical or similar characters of the sequences in the same column. The identical characters of the same column are called matches. So this picture shows you the alignment of sequences A and B showing match characters which are represented here. Match characters are represented non-gray, that means non-shaded, and mismatch characters are represented in the shaded characters. The sequence alignment is nothing but arranging two sequences in such a way to get the matches, to get the maximum number of matches. So here in these two sequences, how many matches we have? 1, 2, 3, 4, 5, 6 matches, and there are 3 mismatches. Moving on further. Whenever we do the alignment of two sequences, there are 4 points that need to be satisfied. What are those 4 points? One more point to understand here. The previous picture where I had shown the alignment of two sequences, there were no gaps introduced, only the two sequences are aligned, and then when they are aligned, we look for the number of matches. In the previous case, we did not see any gaps in the alignment. Why? Because the length of both the sequences are same. When the length is same, there is no necessity for the blanks to be introduced. The two sequences which are being aligned, if they are not of same length, in that case, to get the maximum number of matches in case of the alignment, gaps or blanks can be introduced. So as I told you, what are the 4 points to be satisfied while carrying out an alignment of sequences? Two gaps or blanks which are written as like this, they cannot be aligned against each other. That means in one column, there shouldn't be two gaps, but gaps are introduced in order to get the maximum number of matches. And the second criteria to be satisfied, we can align one symbol or character from one sequence with one symbol or character from the other sequence. That means one character of one sequence can be aligned with the other character of other sequence. It can be match or mismatch. If they are identical, we call it as a match. If they are not identical, we call it as mismatch. And the third criteria to be satisfied, a symbol present in sequence A or in sequence B, they can be aligned with a gap. That means one character in one sequence can be aligned with a gap. Or here, the character in sequence B can be aligned with a gap. So what did we discuss? Two gaps cannot be aligned with each other. You see here, this is the alignment where gaps are introduced in the sequences in order to get the maximum number of matches, because an alignment with maximum number of matches uh, is called optimal or best alignment. So in order to get the maximum number of matches, you can introduce the gaps. But what are we discussing now? There are four different criteria need to be satisfied while carrying out the alignment. So the first 2 points we discussed, two gaps cannot be aligned with each other. You see here, there are no two gaps aligned with each other. That means with each other sequence. We can align one character of sequence A with the another character of sequence B. Two characters from each sequence can be aligned with each other, or one character from sequence A can be aligned with a blank with the uh, uh, can be aligned with a blank in the another sequence. That is possible. Then all symbols present in sequence A, here the complete point is not there. I'll explain what it is. All the symbols are characters present in one sequence, and all the characters present in the other sequence, they all need to be present. You cannot delete them, or you cannot add any extra characters. Sequence A, sequence B are given. You need to align these two sequences in order to produce the optimal alignment. That is the objective. So by following all these instructions with the 4 point, all the sequence characters in sequence A and all the sequence character in sequence B, they should be present. And another thing, they also should be present in the same order. It's not like, okay, just to align, G with G. Here there is no G. Say for example, say for example, what I'm trying to show is, if you don't introduce gap, you cannot, just to get the maximum number of matches, you cannot shuffle the order of the sequence. So all the characters which are present in both the sequences, they should be present, and they also should be present in the same order. But one a slight deviation could be, the position can be changed. What do I mean by position? If this blank is not introduced here, G could have been here. That means G position could have been 1, 2, 3, 4, fifth position. But here, if you see, because of the introduction of this blank, the position of G had been shifted to 6. So what I'm trying to say is, the fourth criteria is that all the characters present in sequence A and all the characters present in sequence B should be present, and they also should be present in the same order, but the position can be changed because of the introduction of the gaps. I hope you are able to follow it. Next, what are the different types of sequence alignments? And one more thing, please remember, whatever the alignment methods or types we are discussing here, they are common for nucleotide as well as protein sequences. The methods are different for nucleotide sequences and they are different for protein sequences. It's nothing like that. These are all the common methods which can be applied for nucleotide as well as protein sequences. So coming to the types of sequence alignment, there are different ways how the sequence alignment methods are classified. One type of alignment is based on the number of sequences that are involved in the alignment. Obviously, if two sequences are involved in the alignment, that is called pairwise sequence alignment, and it is abbreviated as PSA. If there are more than two sequences involved, that alignment is called multiple sequence alignment. So pairwise sequence alignment, there are only two sequences involved and they are aligned with each other. And what is the use of this pairwise sequence alignment? It will help in finding out the regions of similarity or dissimilarity. By finding out the region of similarity or dissimilarity, it helps in finding the evolutionary history of the sequences. So these are the reasons why we carry out this pairwise alignment of sequences. What is pairwise alignment of sequences? The alignment in which only two sequences are involved is called pairwise sequence alignment. Why do we do it? Obviously, to find out the regions of similarity and dissimilarity by which we can understand the evolutionary history of these sequences. So this shows the pairwise alignment of sequences wherein two sequences are involved. Obviously, multiple sequence alignment, as the name indicates, there are more than two sequences involved. So this is a section of multiple sequence alignment of protein sequences where several sequences are involved. Here also, we can find out the patterns of the sequences. By finding out these patterns, we can understand to which gene family this, if they are nucleotide sequences, we can identify the gene family to which these sequences belong to. If these sequences are protein sequences, obviously we can identify the protein family, and it also helps in understanding enzyme active sites and functional and evolutionary relatedness of the sequences that are involved in the alignment. So what are the applications of multiple sequence alignment? Why do we carry out it? Obviously, this also gives us the patterns where the sequences are similar. Those patterns can be identified, which will enable us to understand the gene families. If the sequences are protein sequences, we can identify the protein families, and also enzyme active sites can be identified, and also we can analyze functional and evolutionary relatedness of the sequences that are involved in the alignment. So these are the two types of alignments based on the number of sequences involved in the alignment. And one more thing I just missed here is I was discussing about the 4 points to be satisfied while carrying out the alignment of two sequences, right? So when you satisfy these these four criteria, and when we align the sequences, there is only three possibilities we can see in the alignment. What are those three possibilities we can see in the alignment? In the columns, it can be a match, identical characters can be aligned with each other. This is also match, this is also match. Or it could be a mismatch. So in this particular example, we are not seeing any mismatch, but when a character in sequence A is aligned with another character of sequence B, it need not be all the time the identical characters aligned with each other. If they are dissimilar characters aligned with each other, that is called mismatch. I explained what is match. The identical characters can be aligned with each other, which is called a match. If they are not identical, aligned with each other, in that case it is mismatch. And the third possibility what we would see in alignment of sequences is indel. This indel is nothing but insertion or deletion. What is this insertion or deletion? When the gaps are aligned against the characters, if the gap is present in the bottom row, it is called deletion. If the gap is present in the top row, it is called insertion. So when we align two sequences following these four criteria, we will get the alignment with these possibilities. It could be either match or mismatch or indel, which is nothing but insertion or deletion. I hope this slide I have explained. We can move further. So there are also types of alignment based on the length of the sequences. There are two types of alignment based on the length of sequences: global alignment and local alignment. What is this global alignment? In global alignment, sequences are aligned or compared along their entire length of the sequences. So when the entire length of sequences are included while aligning the sequences, usually for global alignment, the sequences of equal length, they serve as good candidates. What is global alignment? This global alignment of sequences is carried out considering the entire length of sequences. The sequences with the similar length, they serve as good candidates for global alignment. Whereas local alignment, sequences are aligned to find out regions of higher or strong similarity. To find out the sequence regions of higher density or the strong similarity. That means the length of the sequence need not be the same. In the sequences involved in the alignment, they can be of different length. But you will try to look for the regions where there is strong similarity. Uh, Obviously, this kind of local alignment will be helpful when we are trying to find out the active sites of enzymes and genes. For such applications, local alignment of sequences is carried out. I hope you understood the point. Global alignment, uh, it involves the entire length of sequences. So the sequences of the similar length, they serve as good candidates in case of global alignment. Whereas local alignment, the length of the sequences need not be similar. It tries to find out the regions of higher density or strong similarity. We will discuss more in detail about this global and local alignment by discussing the algorithms used for global alignment of sequences and local alignment of sequences in the later classes. But now it is just the introduction part going on. What is sequence alignment? Why is it carried out? What are the different types of sequence alignment? What did I discuss? Depending upon the number of sequences involved, we have pairwise sequence alignment and multiple sequence alignment. Why do we do this pairwise sequence alignment, multiple sequence alignment? I discussed. Then, based on the length of the sequences, we have global alignment and local alignment. Then again, it's kind of repetition here. Why compare sequences? In the very beginning slide, I told sequence analysis is the method where we compare the sequences by the method of sequence alignment in order to find out the regions of similarity and dissimilarity. By finding out these regions of similarity, we can predict the function, structure, and the biochemical nature of these sequences, or obviously we can also establish the evolutionary relationship of these sequences. Then what are these gaps and gap penalties? When I was discussing about different criteria to be satisfied while carrying out the alignment, I mentioned that in order to get the maximum number of matches, we introduce gaps in the alignment. Why we have to get maximum number of matches in the alignment? Any alignment you carry out, at the end, you need to come up with a score for that particular alignment. Higher the score, the better the alignment is. And while scoring the alignments, if there are maximum number of matches, that gets the maximum alignment. That is why while carrying out the sequence alignment, we introduce gaps in order to get the maximum number of matches. So these gaps, when we are introducing, we should have a certain penalties so that we restrict ourselves from introducing many number of gaps. That is why whenever you introduce a gap in the alignment, a gap penalty needs to be included in the scoring scheme which is used or which is applied for scoring the alignment of the sequences. I hope you are able to follow me. Here I'm discussing about gaps and gap penalties. You all know by now, why do we introduce gaps while carrying out the alignment? In order to get the maximum number of matches, right? But we have certain limitations. I'm sorry. We have certain limitations to include the number of gaps. Meaning, if you incorporate a gap in the alignment process, you have to also include gap penalty in the scoring scheme, which is used for the alignment score. Now, one more new word I have used, scoring scheme. This scoring scheme is nothing but a scoring matrix. That means particular number which is defined before carrying out the alignment process, and once the alignment is achieved, based on the scores of the scoring matrix whichever you followed for carrying out the alignment, that needs to be applied, and you have to finally score the alignment score in order to check among the many alignments that are produced for the given sequences, which alignment is the best alignment. Don't get confused. For the given set of sequences, when you carry out the alignment, it's not just one alignment you can produce. There are several alignments that can be produced. Out of all those several alignments, which is the best alignment? How do you decide? By looking at the alignment that has got the maximum score. How do you score the alignments? Based on the scoring scheme which is used for carrying out that particular alignment. So here what I'm saying, when you introduce gaps while carrying out the alignment, you also need to introduce gap penalties in the scoring scheme which is used in the alignment process. There are two different gap penalties. Gap opening penalty, gap extension penalty. What is this gap opening penalty? If you are opening a gap in the alignment, there is no, what do you call that? There is no gap in the alignment. In order to, or while carrying out the alignment, you are newly introducing the gap. In that case, you have to include gap opening penalty. Whereas already existing gap, if you are extending during the process of alignment, there you have to include gap extension penalty. And many scoring schemes include a higher value for gap opening and lower value for gap extension. So what I'm trying to say is gap opening penalty is more than gap extension penalty. Why is gap opening penalty is more than gap extension penalty? That is because it is assumed that multiple nucleotide insertion and deletions are common in nature compared to the single nucleotide indels. That means the multiple deletions or insertions are considered to be more common in nature, which is why the gap extension penalty is lesser than gap opening penalty. So why do we introduce gaps? In order to produce the best alignments having more number of matches. But when we introduce gaps, we have to also include gap penalties in the scoring scheme. Two different gap penalties are used. Gap opening penalty for opening the gap in the alignment, whereas gap extension penalty is for extending the existing penalty. And gap opening penalty is more than gap extension penalty. If you open a gap, you are penalized more, meaning more negative score is incorporated in the scoring scheme. You will have better clarity as we move on further when we look at different scoring schemes or scoring matrices used in the sequence alignment methods. Moving on further. So as I told you, we have to apply scoring schemes when we carry out the scoring alignment methods. There are several scoring schemes available. The simplest scoring scheme which can be used, first we are discussing about the scoring schemes used for nucleotide sequence alignment. So the simplest scoring scheme which can be applied is, if there is a match in the alignment, just give a score of 1. If there is mismatch, give a score of 0. So this is the simplest scoring scheme which can be used for aligning the sequences. If there is a match, give a score of 1. If there is a mismatch, give a score of 0. Different scoring scheme is give different scores for match and mismatches. Meaning, if there is a match, give 1. If there is a mismatch, instead of 0, what we have seen here, give -1. So, for matches and mismatches, give the different scores. This is another simple scoring scheme which can be used for nucleotide sequence alignment. Then the third scoring scheme is give a score for match, definitely negative score, which means penalty for transition, and higher penalty for transversion. In the earlier scoring scheme, if it is a match, we gave the score. And if it is a mismatch, we gave different score, which is a negative score. Here we did not differentiate whether that mismatch is a transition or transversion. I hope you guys know what is transition and transversion. Transition is a, if a purine is substituted by another purine, that is called transition. If a purine is substituted by a pyrimidine, that is called transversion, and it is vice versa. So mismatch could be transition or transversion. So in this scoring scheme, transition and transversion are differentiated. So if there is a match, give a score, positive score 1. And if it is a transition, that means A is substituted with the T, it is not transition, it is transversion. So the higher penalty -2. If A is substituted with G, you all know A, G are purines. So in that case, lesser penalty, that means -1. I hope you understood. This scoring scheme differentiates the transition and transversion, which are two different types of mismatches. So if it is transition, give less penalty, that means less negative score. If it is transversion, more penalty is introduced in the alignment. So these are all different scoring schemes which are used for nucleotide sequence alignment. How about the scoring matrices for protein sequences? In case of nucleotides, we have only 4 different bases ATGC. So the scoring matrix appeared very small matrix. But as you all know, the proteins are made up of 20 different amino acids. So the scoring scheme, whatever you develop, it is going to be 20 by 20. So the scoring matrices for protein sequences, the way we have seen in case of nucleotide scoring schemes, here also the simplest scoring scheme for protein sequences could be give a score of 1 if it is a match, give a score of 0 if it is a mismatch. Simple. So this entire table, which is a 20-20 matrix, is entirely filled with either 0 or 1. So when do we give 1? If it is a match, you give 1. If it is a mismatch, you give 0. This is the simplest scoring scheme that can be applied to the protein sequences. but it does not give enough information about the protein sequences which are involved in the alignment. So other scoring schemes for protein sequences that are available are developed based on observed chemical or physical similarity of the sequences that are involved in the alignment. So what are these physicochemical characteristics of the amino acids which are considered while devising scoring scheme? That could be aromaticity, or it could be charge, or it could be polarity, hydrophobicity, size. These are all the different characteristics, physical or chemical characteristics of the amino acid, based on which, or considering these characteristics, the scoring schemes had been devised. So how the scoring scheme had been developed? In case of protein sequence alignment, a match of identical amino acid residue, if there is a match in case of the identical amino acid residues, that gets the highest score. A mismatch with both amino acid residues, if there is a mismatch and these two amino acid residues, if they belong to the same group of amino acids, group in the sense it could be based on the physicochemical properties. If they belong to the similar group, uh, then it gets higher score. If that mismatch contains the amino acids that have got different characteristics, or in other words, if they belong to the different group, in that case you get the less score. Please understand, I'm telling that the scoring matrix, whatever is shown here, this is the simplest scoring matrix for the protein sequences, wherein a match of, wherein a score of 1 is given in case of the match, a score of 0 is given in case of the mismatch. But this is a very simple scoring matrix which does not give enough information about the sequences which are involved in the alignment. So better scoring schemes had been devised for protein sequences. One such scoring scheme is, which is developed based on the physicochemical properties of amino acids. What are the physicochemical properties that are considered as I listed earlier? It could be aromaticity, charge, size, or what else I told? It can be polarity. So these are all the characteristics based on which amino acids are grouped into different groups. So there is a scoring scheme which is developed if there is match of amino acids in the alignment, that gets the maximum score. If there is a mismatch, and in that mismatch, both the amino acids, if they belong to the similar group, in that case, it gets the higher score. But this higher score is less than the maximum score. If that mismatch includes the amino acids which belong to the different groups based on their characteristics, in that case it gets the least score. So though it is mismatch, depending upon the relatedness between the amino acids, it gets either high score or less score. So considering the characteristics of amino acids, this is how different scoring schemes had been devised. And also, scoring matrices are developed based on the genetic code which is used for protein sequence alignments. So in these matrices which are developed based on the genetic code which is used for protein sequence alignments, what happens here? The scores are given based on the number of mutations that are required in the nucleic acids for changing one amino acid into another. So what I'm saying, the third type of scoring scheme which has been devised for protein sequences is based on the genetic code used by the amino acids. Okay, so you all know one amino acid can utilize different genetic codes, isn't it? That can be encoded by different genetic codes. You all know what is synonymous genetic code. There are more than one triplet codon that codes for the same amino acids. So based on the genetic code information also, by looking at the relatedness of these amino acids, scoring scheme is developed. So here it looks at the number of mutations that are necessary for changing in one amino acid into another amino acid. So based on the number of mutations required, the closeness is decided. Accordingly, the scores are given. In case of the scoring scheme, which uses the genetic code of amino acids. What did we discuss so far? Whatever is shown on the screen is the simplest scoring scheme which is used for protein sequences. But better scoring scheme that considers the physicochemical properties of amino acids, their maximum high and less scores are given depending upon the type of amino acids that are aligned with each other. And the third scoring scheme, it makes use of the genetic code which is utilized by that particular amino acid. All these scoring schemes, whatever I discussed now, they do not give enough information about the evolution of these sequences. They're very simple. Whatever we discussed so far, they're all very simple and they do not give any information about the evolution of these sequences. So a better way that can be used to derive scoring matrices is based on the observed substitution rates among various protein sequences that are present in nature. Please try to understand all the scoring sequences. All the scoring sequences, whatever we discussed so far, are very simple because they do not give enough information about the evolution of these sequences. So the better way to develop this scoring matrix is using the information of observed substitution rates of various protein sequences present in nature. Look at the protein sequences that are present in nature and you see how during the period of evolution, how the substitution, how the frequency of substitution happened. Take that information into consideration and then develop the scoring scheme. That would be the best way of developing the scoring scheme, which will give enough information about the evolutionary relationship of these sequences. So by applying such scoring schemes while carrying out the alignment, you can produce the alignments that have got biological significance. Such scoring matrices, which takes the information of observed substitution rates of several protein sequences present in nature, are called substitution matrices. They are called substitution matrices. I hope you understood the three different ways what we discussed about protein protein sequence scoring matrices are simple. They do not provide any evolutionary information about those sequences involved. So the best way to develop scoring matrix is by taking the information of observed substitution rates of these amino acids among various sequences present in the nature. That gives the alignments which are biologically significant, which means that it gives the information about the evolutionary relationship of these sequences which are involved in the alignment. Such matrices are called substitution matrices. What are these substitution matrices? The matrices which are developed based on the frequency of amino acid substitutions in the protein sequences that are present in the nature. Two such substitution matrices are PAM. PAM is point accepted mutation matrices or percent accepted mutation matrices. And the second substitution matrix is BLOSSOM. This BLOSSOM is block substitution matrices. Block. From the block, BLO is taken. substitution SU is taken, matrix M is taken. So it is abbreviated as BLOSSOM, block substitution matrices. So the SPAM and BLOSSOM are the amino acid substitution matrices, which are very popularly used in several sequence alignment algorithms. So we will discuss more about these substitution matrices in the next class. So just have this information and come prepared for the next class, where we will discuss in detail about substitution matrices. So thank you. That is all I wanted to cover in this class. What did we cover in this class? What is sequence alignment? It is a method which is used for the sequence analysis, which is important application of bioinformatics. By doing this sequence analysis, we can identify the extent of similarity or dissimilarity of the sequences involved in the alignment, by which we will be able to predict the structure, function, and other properties of the sequences. And we also can establish the evolutionary relationship of the sequences. Different types of sequence alignments are there. Depending upon the number of sequences involved, you have pairwise sequence alignment with only two sequences are involved. Multiple sequence alignment, more than two sequences are involved. These are carried out to find out definitely the similarity of the regions, which will help us in understanding the gene families, protein families, or active sites of enzymes, and obviously to understand the evolutionary relationship of these sequences involved in the alignment. And depending upon the length, we have global and local alignment. Then we also discussed about gaps. Why do we introduce gaps while carrying out the alignment? When we introduce gaps while carrying out, we also include gap penalties in the scoring scheme. Then we discussed about scoring schemes. In case of nucleotide sequences, simple scoring schemes are used, like, give scores of 1, 0, 1 in case of matches, 0 in case of mismatches, and give negative scores in case of mismatches, and give different negative scores in case of transition and transversion. Then coming to the matrices for protein sequences, simplest scoring scheme is giving 1 and 0 for matches and mismatches. Then scoring schemes or matrices had been devised based on the physicochemical properties, and the genetic code used by the amino acids also had been used for developing scoring schemes. But all these different scoring schemes, they do not give any information about evolutionary relationship of these sequences, or evolutionary information is not provided by these scoring schemes. So the better way to devise a scoring scheme is by taking the information of observed frequencies of amino acid substitutions of the several sequences which are present in nature. In simple words, by looking at the frequency of amino acid substitutions in proteins, we can come up with a scoring matrix or scoring scheme, by applying which you can produce the alignments that have got biological significance. That means such alignments will give sufficient information about the evolution of the sequences which are involved in the alignment. Two important substitution matrices need to be discussed. One is PAM, point accepted mutations matrix. The other one is BLOSSOM, block substitution matrix, which we will be discussing in the next class.